Every time a cell divides, the machinery copying the deoxyribonucleic acid fails to reach the very edge of the linear chromosome. This unavoidable gap creates a molecular fuse that shortens with every cycle. Deoxyribonucleic acid polymerase requires a ribonucleic acid primer to begin synthesis. When the final primer on the lagging strand is removed, no upstream hydroxyl group exists to allow a polymerase to fill the remaining space. To prevent the loss of vital genetic instructions, chromosomes are capped with long, repetitive sequences called telomeres. These disposable buffers absorb the erosion of replication without sacrificing functional genes. In humans, the telomeric sequence consists of thousands of tandem repeats of the hexamer thymine, thymine, adenine, guanine, guanine, guanine. This highly conserved pattern is the universal signature of vertebrate chromosomal protection. The telomere does not end bluntly. The guanine-rich strand extends beyond its partner, creating a 3' single-stranded overhang that is essential for the structural integrity of the chromosome tip. A specialized group of 6 proteins, known as the shelterin complex, binds specifically to these repeats. This protein shield hides the chromosome ends from the cellular deoxyribonucleic acid damage response machinery. The single-stranded overhang invades the upstream double-stranded telomere, forming a structure called a T-loop. This configuration effectively tucks the free end away, preventing it from being mistaken for a broken strand. After roughly 50 to 70 divisions, telomeres reach a critically short length. This triggers a permanent growth arrest, known as senescence, effectively acting as a biological clock to prevent genomic instability. Certain cells bypass this limit using telomerase, a unique ribonucleoprotein complex. It possesses the rare ability to synthesize new telomeric deoxyribonucleic acid, extending the chromosomal fuse indefinitely.
Telomerase contains an internal ribonucleic acid component called TERC. This molecule provides the template for the repeats, ensuring that the enzyme always adds the correct sequence to the chromosome end. The catalytic heart of the enzyme is telomerase reverse transcriptase, or TERT. This protein uses the internal ribonucleic acid template to catalyze the addition of deoxyribonucleotides to the 3' overhang. The process begins when the telomerase ribonucleic acid template base pairs with the existing telomere overhang. TERT then extends the strand by adding the complementary nucleotides until the template is exhausted. After adding one hexameric repeat, the enzyme must translocate. It detaches and repositions itself further down the newly synthesized strand to begin the cycle again, showing high processivity. Once telomerase extends the leading strand, conventional deoxyribonucleic acid polymerase alpha primase synthesizes the complementary, cytosine-rich strand. This restores the double-stranded nature of the telomere architecture. In most adult human cells, the gene for TERT is epigenetically silenced. Without this enzyme, our somatic cells are destined to age and eventually die, limiting the potential for uncontrolled growth. Approximately 90% of all human cancers find a way to reactivate telomerase. By overcoming the Hayflick limit, these malignant cells achieve replicative immortality, allowing for endless tumor expansion. The most frequent way cancers turn telomerase back on is through point mutations in the TERT promoter. These mutations create new binding sites for transcription factors that drive abnormal protein production. Specifically, these mutations allow the GA binding protein transcription factor to bind. This recruitment forces the cell to pump out telomerase, providing the fuel needed for the cancer to survive forever. Before telomerase is reactivated, precancerous cells often enter a state of crisis. Their critically short telomeres fuse together, leading to catastrophic chromosomal rearrangements that actually drive further oncogenic mutations.
Fused chromosomes create anaphase bridges during division. These bridges snap at random locations, creating massive deletions and duplications. Telomerase eventually stabilizes these damaged genomes, locking in the cancerous traits. 10% of cancers use an alternative mechanism called ALT. Instead of using an enzyme, they employ homologous recombination to swap and extend telomeric sequences between different chromosomes. Cells using the ALT pathway are identified by specialized structures called ALT-associated PML bodies. These nuclear clusters contain telomeric deoxyribonucleic acid, recombination proteins, and shelterin components working in tandem. Since telomerase is nearly absent in healthy tissue but rampant in tumors, it serves as a powerful biomarker. Detecting telomerase activity can help clinicians identify early-stage malignancies with high precision.
The telomeric repeat amplification protocol, or TRAP assay, is the gold standard for measuring this activity. It involves extending a primer with telomerase, followed by polymerase chain reaction to amplify the results. Immortal is a potent inhibitor that targets the telomerase ribonucleic acid template. By binding to the template sequence, it physically blocks the enzyme from recognizing and extending the natural telomere ends. Guanine-rich sequences can fold into stable four-stranded structures called G-quadruplexes. These knots are natural barriers that telomerase cannot easily unwind, effectively shutting down the elongation process. New therapies aim to stabilize these G-quadruplexes using small molecules. By locking the telomere in this folded state, the cancer cell can no longer maintain its immortality and eventually enters senescence. Another strategy involves vaccines that prime the immune system to recognize TERT peptides. Since cancer cells overexpress the protein, cytotoxic T lymphocytes can be trained to hunt and destroy these immortal cells. Not all telomere problems lead to cancer. Inherited mutations that reduce telomerase activity cause telomeropathies. These diseases are characterized by premature aging of tissues that require constant cell division. Dyskeratosis congenita was the first recognized telomeropathy. Patients suffer from bone marrow failure, skin hyperpigmentation, and nail dystrophy due to their inability to maintain sufficient telomere length in stem cells. Short telomeres are also a major risk factor for idiopathic pulmonary fibrosis. The exhaustion of lung stem cells leads to abnormal scarring, making it difficult for the organs to exchange oxygen. Telomerase is stabilized by a protein called discarin. It binds to the H/ACA box of the TERC ribonucleic acid, protecting it from degradation and ensuring the complex is properly assembled. Recent breakthroughs in high-resolution cryo-electron microscopy have revealed the atomic structure of human telomerase. We can now see exactly how the protein and ribonucleic acid subunits interact in 3 dimensions. The TERC component contains a vital pseudoknot domain. This complex folding pattern is essential for the catalytic activity of TERT, as it helps position the template correctly within the active site. Beyond sequence mutations, TERT is regulated by histone modifications. Acetylation of histones around the TERT promoter opens the chromatin, allowing transcription machinery to access the gene and produce the enzyme. Telomerase is not always active in the nucleus. It is dynamically trafficked between different compartments. During most of the cell cycle, it is sequestered in Cajal bodies before being recruited to the telomeres. Telomerase recruitment occurs specifically during the S phase of the cell cycle. This timing ensures that telomere extension happens in coordination with the overall replication of the cellular genome. The shelterin protein TPP1 acts as the recruiter for telomerase. It contains a specific surface called the TEL patch that interacts directly with TERT, pulling the enzyme onto the chromosome end. The balance between enzyme processivity and the frequency of recruitment determines the final length of the telomere. Subtle changes in this equilibrium can mean the difference between healthy aging and disease. Recent evidence suggests TERT has non-canonical roles outside of telomere maintenance. It can move into the mitochondria to protect against oxidative stress and even influence gene expression as a transcription factor. Inside the mitochondria,
TERT binds to mitochondrial deoxyribonucleic acid and improves the efficiency of the electron transport chain. This suggests that the enzyme's impact on longevity is multifaceted.
Targeting telomerase is a delicate balance. Inhibiting it could kill cancer cells, but might also damage healthy stem cells. Conversely, activating it to treat aging might inadvertently increase the risk of developing tumors. To solve this, researchers are exploring combination therapies. By using telomerase inhibitors alongside traditional chemotherapy, doctors can force cancer cells into a vulnerable state that is much easier to eliminate. Cancer cells are masters of evolution. If telomerase is blocked, some tumors can switch to the ALT pathway to maintain their telomeres, highlighting the need for multi-targeted therapeutic strategies. In the future, controlled transient activation of telomerase might allow us to rejuvenate aging tissues. By briefly turning the enzyme on, we could potentially reset the cellular clock without triggering oncogenesis. Stem cells naturally express moderate levels of telomerase to maintain their self-renewal capacity. Understanding how they regulate this level perfectly could provide the blueprint for safe anti-aging interventions. Advanced liquid biopsies are now being developed to detect telomerase messenger ribonucleic acid in the bloodstream. This non-invasive approach could revolutionize how we monitor cancer recurrence after surgery. The silencing of telomerase in somatic cells likely evolved as a potent tumor suppressor mechanism. Humans traded the possibility of individual immortality for a lower risk of early life cancer. Chemists are now creating synthetic telomere mimics that can bind to the ends of chromosomes and perform the protective duties of natural telomeres. These artificial caps could one day bypass the need for enzymes. Telomerase remains the central pivot in the battle between aging and cancer. By mastering this enzyme, we hold the key to either stopping the immortal growth of tumors or extending the healthy human lifespan.